Hello, thank you for inviting me to give this talk about the cannabinoid receptors, and I'm going to focus on the CB2 cannabinoid receptor and the relation on emotional behavior, especially anxiety and depression. You all know about the cannabis plant and about the different type of cannabinoids, more than 120 cannabinoids. We now also we know about endogenous cannabinoid, anandamide, to academic glycerol, to AG, and some others. And we also know different type of compounds that are going to act, activating the receptors or blocking the receptor, like all of this. I ask you just to concentrate on this one, that I'm going to use it later, the AM630, that is a cannabinoid CB2 receptor antagonist. We have two cannabinoid receptors, and I'm going to focus on this one. The CB1 receptor was first described in this sagittal section of the laboratory of Miles Herkenham in 1990, and you know that it's located in many different regions: frontal cortex, hippocampus, caudate putamen, globo pallido, centropeduncular nucleus, substantia nigra, hypothalamus, maybe in the ventromedial nucleus, amygdala, and several layers of the cerebellum. About the CB2 receptor, there was a lot less information. We came to 1983, just showing that the CB2 receptor was in spleen and in this other paper on lymphocytes. So it was thought that the CB2, because we were not able to measure it in the brain, was a peripheral receptor, not in the brain. On 2003, on the different situation, not on the basal condition, but on the brain pathological condition, it was found for the first time in this situation, overexpressed in neuritic plaque in Alzheimer's disease brain patients of Alzheimer. And from here, it was also found in some different pathological conditions, for example, in monkeys with meningoencephalitis, close to brain tumors, and in some other situation. So in 2005, it was published for the first time that the CB2 was present under basal condition in the brainstem of rat, mice, and ferret. What we did is in 2006, it was described also in the rat brain, the protein and the gene also distributed in the same different areas than the CB1 receptor. So we built a CB2 mouse, a mouse overexpressing the CB2 receptor using a prionic protein, and after several years of different tests, we came out with one of these transgene, and we described first in how was the gene expression of the CB2 under basal condition in a wild-type mice. And as you can see, we used a punch technique ligated with RT-PCR, so we were able to do it in many different small areas. And then we did it also in the transgenic mice that is overexpressed, as you see, in all these areas several times. We check also in the hippocampus, and we look at the protein, and the protein is higher in the different layers of the hippocampus. And when we did an immunohistochemistry, you can see that there is an increase of the CB2 in the CB2 XP mice. And we also check in the CB2 knockout mice, and you can see that there is almost no signal. So it appears that this is a good control of our antibody. We also studied the co-localization of the CB2 receptor with neurons in different areas, like nucleus accumbens and VT8. You can see it here, neurons label, and the co-localization here in different areas. And the same thing, we did it for astrocytes also, and you can also see in similar regions that there are co-localization in astrocytes. The first thing we did is we studied the behavior of the animals. We used the open field just to be sure that there was no problems related with the motor behavior. So we're going to be using different type of tests. We used the spontaneous motor activity record tracking, and we found a small increase in certain distance and small decrease in peripheral distance, although the total distance it was the same. See, this is probably a suggestion that these animals may be having less anxiety than the wild type animals. So we performed different tests like dark box and elevate plus maze, and we found that the mice overexpressing the CB2 receptor have less anxiety. They spend more time in the illuminated area, in the lighted area, and also more time in the open arms, also significant of less anxiety. When we did that in the knockout mice, we found exactly the opposite results, less time in the light area and less time also in the open arms, very significant. So this is exactly the opposite than in the mice overexpressing CB2 receptor. Then we went to check about different behaviors related with depression traits, and we used tail suspension and novelty suppressed feeding test. You can find here that the time of immobility of the behavioral despair is a lot smaller than in the wild type animals. They are like more antidepressant, and the novelty suppressed feeding test, the latency to go to eat after being fasted for almost a day is less in the animals overexpressing CB2 receptor, and the consumption of pellets is much, much higher. So after that, we did also tail suspension in the knockout mice. As you can see, here's just the opposite. Time of immobility increased. In the novelty suppressed feeding test, the latency increased, the consumption decreased really very much. Just to be convinced that this was also working in a model of depression, we use a chronic mild stress using different type of stimuli 3 times a day for several weeks with this type of a schedule for 7 weeks. And something that we did a little bit different than many other studies is that we check for the different effects on behavior at different times over this schedule of 7 weeks. We use light dark box, elevate plasmase, tail suspension, again like dark box, and consumption of saccharose. And these are the results in the wild type mice. At the beginning of 2 weeks, we're beginning to have some anxiety that is increases on week 4 and is very, very evident on week 6. On the mice overexpressing CB2 receptor, there was no effect at all for 4 weeks, and there was a small effect in 6 weeks. So what we found here on week 5 is, as you can see, is an increase in the time of immobility. In the wild type, there is no effect on the mice overexpressing CB2 receptor. On week 7, there is a decrease on consumption of saccharose, and there is no change on the mice overexpressing CB2 receptor. So with this in mind, what we try to study is what is this happening? Probably we don't know the whole answer, but we look at the BDNF, and we found that the BDNF decreased after the chronic mild stress. We all know that. It has been published several times, and the immunoreactivity too. In the mice overexpressing CB2 receptor, BDNF is much, much higher than wild type, and there is no effect at all with the chronic mild stress. Remember, 7 weeks of continuous stress. And we can see this on the immunohistochemistry that is here down on the slide. So what we did is, what were the mechanisms that we can use therapeutically using this finding? And we decided that if we give an antagonist continuously at certain dose for a certain period of time, we will increase the number of receptors, and this is what we did. This is the same type of experiment, other than in this occasion, on week 4, we control in wild-type animals these effects, and then we begin the treatment with the antagonist, the one I mentioned at the beginning, AM630, every 2 hours intraperitoneally for 3 weeks, and we measured tail suspension on week 7 and consumption of saccharose 1% on week 8. So this is the same development that we did before on wild-type animals. This is the first week. Second week, we're beginning to have some effect on anxiety. On week 3, the time of immobility increased very significantly, and in week 4, the consumption of saccharose decreased. We have a model of chronic mild stress. So up from here, we use the treatment with AM630, and these were the results. On week 7, 3 weeks of treatment with AM630, the results were block completely in the time of immobility. Here, you just see the difference, and the same thing happened with the consumption of saccharose that increased very significantly in the animals stress treated with AM630. So that means that the AM630, the increase of the receptors, we don't know yet about the increase of the receptor, but the treatment with AM630 is having an antidepressant effect. And now the question is, are we having changes in BDNF to have this type of behavioral effect? Are we having indeed increase in CB2 receptor with depth of treatment? Well, the answer is in the next slide. We measure the CB2 receptor is decreasing chronic mild stress and is increasing with AM630. So indeed, the behavioral action of AM630 at least is related with the increase of the CB2 receptor, and the same thing happened with the gene expression of the BDNF. Look at this enormous, huge amount of increase of BDNF with this type of pattern of administration in the gene expression and also in the immunohistochemistry. So we published this paper and some others just showing that part of the action of the CB2 receptor antagonist may be related with GABA-A and alpha-2 and gamma-2 receptor subunit. And finally, we went to study that in humans, and we used a suicide as a model. So we have an agreement with a different type of organization to carry out this study with different ethical committees. We began the study in 2008 to present, so almost 15 years now. Male Caucasian, subjects never diagnosed of psychiatric disease, and also subjects not taking any drug acting on the central nervous system. So we also took care of different type of parameters, not only the postmortem interval and the pH, but also the postmortem to refrigeration to stop the autolytic process. It's about 3 to 6 hours, and these are the results that we got. We use also the RNA integrity number using the bioanalyzer and doing electrospherograms just to check not only for the value of the RNA integrity number, but also for the 2 peaks of the 18S and the 28S to be intact, which give us also the information about that the RNA is in perfect shape to interpret changes in gene expression. And the results we got is that in the dorsolateral prefrontal cortex of suicide victims, there is a 35 decrease in gene expression, and in the amygdala is about 40% of gene expression. So the CB2 receptor in the brain is also related with anxiety and depression, at least in humans. We also did studies with GRP55, and we were able to show that there are heterodimers of CB2 GRP55 in the dorsolateral prefrontal cortex, and that these heterodimers are located in neuron, in the astrocytes and microglia. So with this, the conclusions are that the mice overexpressing CB2 receptor exhibit a behavioral endophenotype resistant to different type of stressful stimuli. Overexpression of CB2 receptor alters the response to stress of different gene targets involving emotional regulation, such as CRF, POMC, BDNF, GABA alpha-2, and gamma-2. I didn't show all of these results in the presentation. The blockade of the CB2 receptor by AM630 produces anxiolytic and antidepressant action associated to changes in the gene expression of the CB2 receptor and in targets regulating emotional behavior like GABA alpha-2, gamma-2, and BDNF. CB2 receptor and GRP55 gene expressions are significantly reduced in dorsolateral prefrontal cortex of suicide victims, and increase in the heterodimerization of CB2 GRP55 was found in neurons and astrocytes of the dorsolateral prefrontal cortex. And taken together, we believe that these results point out that the CB2 receptor deserves further attention as a new interesting target for the treatment of neuropsychiatric diseases presenting anxiety and depression. And many thanks for your attention.